But I can tell you just the reason why we chose the two of those. We
Hmm.
chose them because, first of all, they've both been used in clinical trials of patients with affective disorders. So, psilocybin has been used in depression, and MDMA has been used for PTSD. And so we thought that these would be clinically relevant, therapeutically tractable compounds to work with.
Yeah, yeah.
And yeah,
Yeah, so it's two different substances. Psilocybin from magic mushrooms, that's, yeah, the classic hallucinogenic psychedelic compound from mushrooms. MDMA, certainly strongly psychoactive, but most people don't really call it hallucinogenic
yeah,
or psychedelic in the so-called classic sense. But then again, if we bring up Gould Dolan's work, part of what she showed is despite the fact that these are different drugs with different effects that act through different mechanisms, they actually have some convergent mechanisms downstream that tie into this metaplasticity idea. And
exactly.
so I think from that standpoint, you would expect them to have similar effects at some level. And okay, so you used those two drugs. What exactly did you do for the main experiments here? Can you describe them in a basic way?
Yeah, so again, I think tying this all back to the kind of the neuroimmune interactions mechanism, what we really did was we subjected mice to a chronic psychological stress paradigm, where we did something called chronic restraint. So we restrained them in a small space for longitudinally. So we did this for basically 1 to 3 weeks, and we were looking for a time point where the mice showed dysregulated behavior. We focused on fear, as well as dysregulated immune responses, and and so we settled on this paradigm that's approximately 3 weeks in duration, where the mice are taken in and out of the tube every day. They spend about 6 hours a day in this tube, and they get stressed out. And this was the time point where we detected these kind of convergent immune and behavioral changes. So that's sort of what we focused on.
So what are the behavioral changes? Are they no longer, are they extra fearful? Are they not responding in the normal fearful way?
Exactly. So the mice essentially are extra fearful. So if you do a classical fear conditioning and then you look at how the mice respond in essentially a new context, the mice exhibit greater fear, let's say generalization, if you wish. And we validated this using, kind of two independent paradigms. And so from there, we were hypothesizing that perhaps the fear center could be, which is the amygdala, could be one of the dysregulated circuits.
Mhm, mhm. And then how is the immune system dysregulated here? What does that look like in these animals?
Yeah, so initially we just did something very simple, which is we surveyed cytokines in the plasma, and it's really just looking for kind of changes in these molecules, which other labs have shown occurred in response to chronic stress. I mean, or even acute stress. So this is work by Scott Russo or Phil Swirsky, a lot of people showing that there's kind of these massive changes in cytokine production or leukocyte shifts. But also there's changes in tissues across the body that produce these cytokines. And so this was sort of just a first approximation, but we wanted to get at how those signals might be actually influencing the brain. And so that's kind of how we got to the time point where we saw immune changes. But then we looked we basically just surveyed the abundance of different immune cells across different tissues in these stressed mice, trying to figure out which specific subsets would be changed in different compartments.
Mhm. What were some of the largest particular changes that you saw?
Yeah, so what was interesting is we actually saw within the meninges, there was recruitment of multiple different classes of immune cells. One of the populations that we focused on is something called a pro-inflammatory monocyte. And these are cells that are pretty short-lived, but they are very important in modulating tissue inflammation. And they also kind of come in different flavors, but this particular type of cell is known to traffic to tissues. It can either circulate out, it could die, or it can also differentiate into a more long-lived type of macrophage. And so they're in circulation until they sort of encounter a tissue, which could be something like the meninges.
Hmm, hmm. I see. So they're induced by some stimulus. depending on where they are and exactly what's happening, they might sort of be around for a little while and then go away, or they can change into a different cell and stick around for extended periods of time.
Yeah, exactly. And these cells are thought to be liberated from the bone marrow in response to, or other tissues in response to chronic stress. And it kind of really depends on how long the stressor is in terms of where these cells go, what they do, and so on.
And so are monocytes, are they like white blood cells that would eat pathogens, or like what do they do functionally?
Yeah, exactly. They're kind of recruited to sites of wound healing, if you wish.
Mm-hmm.
And there, the kind of the tissue instructs them on what to differentiate into, where they can kind of take on more of a reparative role. If there's a pathogen or something like that, they can also secrete kind of these innate cytokines, or also, and also differentiate into, let's say, like types of macro tissue resident macrophages or things like that.
I see. So they're
So
almost like
yeah,
they've almost got kind of like a stem cell vibe where they can become different things depending on the conditions. They might clean up debris, they might deal with
exactly.
pathogens, whatever.
Exactly. Yeah,
Hmm.
that's right.
So how do we get into the brain here?
Yeah, right. So one of the things we began doing is we were looking for basically once we settled on the amygdala, we were starting to look for changes in the brain associated with these time points where we saw kind of, let's say, convergent neuroimmune interactions.